Hello, everyone. Thank you for attending this webinar. And we're going to talk about tiny biosensors and the biochips and their biggest potential in healthcare. So there are two ways to rationalize why this is a very important topic. First, it's application-driven. And in U.S., we know that healthcare cost is exploding and the population is aging rapidly. And that's true for many advanced countries shown here. And the U.S. is kind of an outlier because we spend more money per capita than any other country in the world. And the total healthcare spending is $2.5 trillion in 2013. And we can anticipate that this trend will also show up, similar trends will show up in developing countries as those countries also become more advanced and people live longer and longer. And when you look at the healthcare spending versus age, you find that the spending after 50 years old is much more than when you're younger. And that's quite normal because we usually don't get sick very often when you're in 20s and 30s. But this curve is, the red curve for U.S. is actually quite alarming, and this means we're spending money where it's not most efficient. And we anticipate that in the future, we can spend more money early to keep people more healthy with a better life style and have more technologies to be well, and then we can actually reduce the spending burden in later stages of life. So that's where technologies will come in. Regardless of the applications, we are inventing many technologies in both biotechnology, medical technology, and information technologies, many other fields. And here I just list a few: nanosensors, genome sequencing, wearables like smartwatch, Fitbit, Jawbone, etc. And this chart shows the cost of sequencing a human genome versus a year. And the purple curve is a famous Moore's law in semiconductor industry. And the green line shows the cost per genome sequenced. And you can see initially it's dropping just like Moore's law, but in recent decades, the cost per genome has dropped precipitously, and now we are at about $1,000. And that opens a lot of application avenues in healthcare. And there are many other such inventions. And these, once properly connected to a right type of application, it will be game changers in healthcare arena. So from this slide forward, I will show you a few examples of tiny chips which can have greater potentials, and some of which has already made big changes in healthcare. The first example is real-time in vivo biosensors developed by Professor Tom So, who's just joining Stanford faculty. He previously worked at the UC Santa Barbara and he actually graduated from Stanford Electrical Engineering Department, and so it's a homecoming for him at Stanford. We're very pleased to have him back on board. And his group has developed this amazing in vivo biosensor shown here. In B, that's basically a schematic of the sensor. And it's basically an electrical chemical biosensor. And the electrode is typically gold. And on this gold, we immobilize an aptamer. And this aptamer is labeled with a chemical in the end. And this chemical, when sees certain molecules in solution, they will go through chemical reaction and produce electrons. And if this aptamer is just like shown in the left, these electrons will basically dissipate in the solution and move away. Now, if there are certain molecules in the solution, usually it's a molecule of interest, often called analyte. And this aptamer will then fold, and at the end of the aptamer, this blue dot will come very close to the electrode. Now, if you induce the electrochemical reaction again, these electrons will be very close to the electrode, and they will be picked up by the electrode. So now you have a much more pronounced electro signal detected. So that means the electro signal now will have a one-to-one correlation to the concentration of green analyte here. And that's the basic principle of this real-time in vivo biosensor. And why is this technology game-changing? Because Dr. So's group has made this sensor so small, they can implant it into an animal right now, and in future, in human. As we know that we take a lot of medicines when we get sick, and different people will have different dose to see efficacy. And right now, doctors don't know which person has the best dose to use. And if you use too much medicine, oftentimes they have toxicity. Okay, so there's a window of best dose between efficacy and toxicity, and that varies from person to person. So with this biosensor, you can actually measure the dose of drug inside the body circulating in real time. And in this Science Translational Medicine paper published, you will find that Dr. So's group has managed to measure doxorubicin, a common chemotherapy drug for cancer, and they can reliably monitor the dose change of the doxorubicin inside the body. Another example of tiny biochips is from my own group. Shown in the middle here is a device called mega Sifter or magnetic sifter. They are fashioned out of a silicon substrate with silicon nitride membrane, and we etch small pores into the silicon nitride membrane. And after that, we coat the membrane with magnetic films. If you apply a magnetic field to this device, the magnetic film will be magnetized and generate a very strong magnetic field gradient. So if there are any small magnetic objects passing through, they will be captured on this device. So what's their use? Well, they are very powerful tool for capturing so-called circulating tumor cell, also called CTCs commonly in the field. As you know, when a patient get a cancer, they usually originate in one place. So if it's a lung cancer, it's usually originated in lung. But the lung cancer itself does not kill a patient. Usually, the cancer will grow and eventually it will metastasize. And many other secondary cancers will pop up everywhere, and then the person will not be able to handle it anymore, and will give way to the cancer, and that's the tragic end of the disease progression. Now, the key to diagnose cancer and also to cure cancer in the future is to study CTCs. But the CTCs is very rare in blood circulation. For 1 billion healthy normal cells circulating in blood vessel, there's about 1 to 100 CTCs. Okay, so it's truly searching a needle in a haystack when you try to enrich and capture CTCs. So this is a tube of blood sample, and we take this from the hospital and then mix this blood sample with magnetic nanoparticles. So the green cells are circulating tumor cells, and they are labeled with a black dot, so now they're magnetic objects. All the other dots, red and gray, are healthy cells like white blood cell, red blood cells, etc. So you take this mixture, pass this device, and all the unlabeled healthy cells and other matrix components in the blood will pass through, and the labeled CTCs will be captured. After that, we turn off the magnetic field. Now we can release magnetic-labeled circulating tumor cells. And then we pass to a device called nano-well. This is made up by microfluidic technology. And each well will have one or no CTCs. And then we do chip-wide reverse transcription polymerase chain reaction, or so-called RT-PCR. Basically, we amplify RNA molecules in each well. And from that, we can study mutations associated with CTCs and various gene expression patterns in the cells. So from those signals, we can identify CTCs further, and we can study their mutation status. And this can be used for cancer diagnosis and also to inform doctor what drug to use for a particular patient. Now, this is a slide from Google, and Google X has a vision to develop so-called wearable disease diagnostic. The idea is that you have a wearable device on your smartwatch or smart band, and those smart bands have a device, an optical sensor, for example, to see objects flowing in your bloodstream. And one vision is that if we, say, wanted to detect circulating tumor cells in blood, in the last slide, we have to draw the blood and take outside of the body to do so-called in vitro diagnosis. In this case, we don't take any blood out of the body. Instead, we inject nanoparticles or nanolabels into the bloodstream. they're biocompatible, so they will not cause harm to your body, but they will latch on to circulating tumor cells. Now these circulating tumor cells are labeled by these orange nano labels, and they will emit certain signals, say optical signals or other signals, and they will be picked up by the smart band. And now, without blood draw, without going to hospital, you may get early warning of cancer onset. And this will be the future technology. And I hope to hear good news from Google X or other parties in the future about this fascinating vision. In my own group, we are also working on mobile biosensors. In this case, we use a magneto nanosensor, which is buried in the bottom of this nano well. And this is a tiny PCB board, so we can talk to the sensor as the sensor detects the analytes. I don't have time to go over the details of this sensor. You can certainly look up my papers in the internet, but here is a very quick demo to show that with this sensor we can detect antibodies against hepatitis B. So to analyze whether you're protected against hepatitis B virus or not. Okay, so this is a real-time video, and it's sped up at a certain portion. So the whole thing in real time lasts about 15 minutes, but it's fast-forwarded to allow you to see the whole process. And here my student is doing a finger prick to draw a tiny drop of blood and mix with some buffer solution and then load it to this reader station. And the reader station is completely controlled by a smartphone, and the sensor is already inserted from the side here. Now the test is finished. The whole test itself took about 10 minutes, and the test results now is emailed to the medical record of your primary care. And this is a test result for 10 volunteers. And each bar associated with the concentration of antibody against the hepatitis B measured. And you can see that for volunteer 1, 6, and 8, their antibody level is very low. So by WHO standard, these people are unprotected. So if they have exposure to hepatitis B virus, they may become sick. Now, for other volunteers, they have high level of antibody against hepatitis B, and they will have very low probability being infected by this virus. And I happen to be one of the volunteers and one of the 3 unprotected ones, and I'm hoping to see my doctor to get a boost shot of antibody, vaccine, to get my antibody level up so I can be safely traveling in East Asia and Africa. Those are areas that have high prevalence of hepatitis B. We also keep track of many other emerging biosensors in other universities and companies. Another example here is an optomagnetic biosensor from Philips, which is specifically designed for application at the point of care. And this is a video from Philips' website. I'm obviously not a paid endorser for Philips. This is shown to you just to illustrate the many exciting tiny chips out there. And this biosensor is exciting to me because it can be used to detect a heart attack at a point of care. And on the left is data published by Philips in 2009 article on Lab on Chip. And they use this device to measure cardiac troponin, CTN is cardiac troponin, and this is a signal versus concentration. And you can see that they can detect the signal down to about 1 picomolar or about 24 picogram per milliliter. So this is very high sensitive biosensor. And for a healthy person, normally the cardiac troponin level should be very, very low. But if there's a blood clot of some sort in the artery of heart, here shown number 1, the downstream heart vessels will be cut off blood supply and depleted of nutrition, and so the muscles will start to die, and those processes will generate cardiac troponin, and they will start circulating in bloodstream elsewhere. And so if you have a sensitive biosensor to detect the onset and increase of cardiac troponin, then that's you can detect a heart attack early and save lives. And I hope that in the future, this kind of device will become a wearable device on people who need it. Another technology I wanted to talk about is bioelectronic medicine. And this is a homegrown technology in Professor Ada Poon's lab at the Stanford Electrical Engineering Department. This is a device shown at top, and blue lines are nerve systems in human body. And if you insert this device at the right spot, right nerve of the brain, basically you can reduce the symptom or prevent even things like epilepsy or depression just by giving a tiny electrical pulse at the right nerve spot. And this is a very exciting area, and I'm sure you will hear more about this technology from Stanford and many other research organizations. So this brings me to my last slide for my portion of the presentation. We are developing tiny biosensors and bioimaging tools, which was supposed to be Professor Adam Delazorda's talk, but you will probably find him elsewhere in the future. All these technologies will allow us to achieve so-called data-driven diagnostics and medicine. And that will, of course, translate into better diagnosis, more effective therapy, and new electronic medicine, in addition to drugs, etc. And that will allow us to really make technology work for humankind. That's our goal. And we use technology to study the entire human body, the cells in human body, proteins, antibodies, DNAs, RNAs, some of which I touched in previous slides. And I hope that I will meet you on internet again in the future.
Thank you, Shan. Great, let's get into some of the questions.
So the one question is, any biosensors to detect a stroke, brain attack, or heart attack? So the troponin or cardiac troponin, which I talk about, is a biomarker for heart attack. And so obviously the sensor from Philips is designed to detect heart attack and many other technologies. For stroke, which happens in brain, unfortunately, there's no such biomarker equivalent to see cardiac troponin or CTN, and but there are many people working on that, and in the near future, I think such technology will be available. These sensors FDA approved. The in vivo biosensor from Dr. So's lab is not FDA approved. As you recall, so far the technology has only been applied to animal because it requires chemo drug injection, and you really can't do this kind of test very easily in human. But they are moving to human, and hopefully in the future that it will be FDA approved. And the Philips optomagnetic biosensors, I believe, is approved in Europe, but it will probably be approved in U.S. in the future if things go well. So the next question is, were these new technologies available on market? The optomagnetic biosensor from Philips is available on market. The other technologies I mentioned are all in the research development stage. They are not publicly available on market at this point. So the next questions that I mentioned, I hope would the Philips optomagnetic biosensor will be made wearable. That's an impromptu comment. I do not know for sure if Philips is working on a wearable version of that device, but I think that's a comment from application pool perspective, because we often hear stories like someone went jogging and got a heart attack in the middle of the jogging, and by the time it was discovered, it was too late to be saved. And so if we have a wearable device like Apple Watch, like a Fitbit, and it could measure the tiny amount of troponin in bloodstream, then before jogging or in the beginning of jogging, you start to see this upregulation of biomarkers, and you can, or instead of go for a long jog, you jog around the hospital or just cancel the jog that day and watch for your other vital signs. So I think that kind of device will be much more powerful than just a device to measure your heart rate or your body temperature. Okay, so the next question is that the possible harm of using mobile phones and other EM devices, and also the biodegradable sensors. As you know, there's no free lunch. So in healthcare, whenever we introduce a new drug, a new device, to various degrees, there are some cons to balance out the pros. And that's where, the engineers and the researchers come in. We have to understand pros and cons of every new technology, and then we can deploy them intelligently. And so for biodegradable sensors, we have to make sure, what's the degree of the side effects. And so if there's certain side effects, we probably should not market those devices to everyone. If you are reasonably healthy, you probably shouldn't wear this, troponin wearable device. But if you have family history of heart attacks and you have very high level of cholesterol, you have high risks of a heart attack, then you probably should spend the money to buy this wearable heart attack device, or in the future, stroke sensors. Okay, the next question is, could you speak to the economics of nanosensors for emerging markets? And also, what's the coefficient of variation for these sensors? Okay, emerging markets is a bigger market, even though they're emerging for a lot of biosensors. And unfortunately, relative to the OECD countries or advanced countries, people have less purchasing power. So all these new technologies being developed would have a harder time to be economically successful in emerging markets. So a cancer test, which is will allow the developer and the companies to make a profit in U.S., not necessarily in China or India. So that's the reality of anyone who's planning to develop new technologies in healthcare area. In terms of coefficient of variations, that's a very loaded question, and I really can't answer about them for all the technologies, but just two examples. The Sifter device from my own lab, which I talked about, that has a coefficient of variation of 10% to 50%. So it's quite a variable. So it's really not a good device for just count how many CTCs which are there in each person, because the technology is not really precise in terms of counting CTCs. But it's a very good technology for catching CTCs and study what kind of mutation happened in cancer. So for one mutation, like a EGFR mutation in lung cancer, then there's certain so-called target drugs can work very well for that mutated cancer cell. So in that particular application, that device is very, very useful in spite of large coefficient variation. Now for the optomagnetic biosensors from Philips, the coefficient variation I think is showed in one of the standard curves, and those devices I think can be made to have CVs below 20% and meet FDA requirements in the future.
Thank you very much for all of your time today and effort. This has been wonderful. I learned a couple new things that I'm very excited about. This I didn't know that technology, we always think technology is advancing too quickly, but these are some great advancements. For more information on any of the online graduate courses, please check out scpd.stanford.edu, and you can get more information on enrollment requirements and course information as far as when they'll be offered again. Thank you very much.
Allow me to thank you, Stacy, for hosting me. This is very exciting seminar for myself. And also I want to thank everyone who dialed in and asking so many great questions.